So I want you to all now, the alums in particular, just think for a moment. I want you to think about the faculty member at Tufts that had a big impact on your life. And I bet most of you can think of that person. Tufts has done a great job over the years of recruiting and retaining fantastic faculty. And our goal is to be sure that in 20 or 30 years, when today's students and future students are sitting in these seats, we have outstanding faculty who can be the people they're thinking about at that point. And I'm excited today to introduce you to one of those faculty of the future, Ben Wolfe. Ben joined the biology department in arts and sciences in 2014. He has a PhD in 2011, so relatively recently. He does fantastic work. His lab, as you'll hear today, uses fermented foods as a model to identify the mechanisms that drive the ecology and evolution of microbiomes. Findings from his lab help develop design principles that can guide the manipulation of microbial communities in agriculture, industry, medicine, and nature. He's also a faculty member who epitomizes what it means to be a student-centered research university. And you'll hear today not only from Ben, but from a former student of his. Ben?
So we're not alone tonight. I know many of you have come with friends or colleagues, but you also came with your microbes. So as I was watching you come in, I was thinking to myself, there's another 1 billion bacteria coming in the door, another 1 billion bacteria coming in the door. So everyone in this room has a microbiome. You've probably heard about the microbiome in the news. It's a very hot buzz phrase in medicine and in biology in general. What is this thing called the microbiome, that community of microbes that's living in and on your body? Well, it's simply the bacteria, the fungi, the viruses that are living in a particular habitat. So you hear a lot about the human microbiome, and specifically the gut microbiome, but everything has a microbiome. The carpet in this room, for example, for better or worse, has a microbiome. New York City has a collection of microbes that live inside of it and we can consistently find. That is the New York City microbiome. One really interesting fact when you think about the human microbiome and just how important microbes are to your life, if you count all the cells in your body, half of those would actually be human cells and the other half would be bacteria. So you're half microbe. And so this idea of the microbiome isn't new, but what's happened is new DNA sequencing technologies, largely from the Human Genome Project, have rapidly opened up the black box of microbial diversity. So about 15 years ago, you couldn't figure out how many microbes were living in the soil just outside the door right there. But today, I can do that in 2 days and tell you about the thousands of microbes that are present there. What we're learning very quickly is that microbiomes are essential for life. So out in natural ecosystems, from oceans to Arctic soil to rainforests, microbes are turning over nutrients, they're feeding plants and animals, and they're killing things but also keeping things alive. In our food system, there's a lot of interest right now in understanding how microbes impact productivity, safety, and quality of food systems. So on the roots of a corn plant, for example, there are many fungi that are making that corn plant healthier and able to produce more corn. And again, back to your own body, we're learning every day about the interesting thing that microbes are doing to your human health. So for example, as you're developing as a youngster, microbes in your gut can fine-tune your immune system and affect the way that you respond to pathogens in the future. Now, this is all very exciting. The microbiome's super hot and super exciting, but there's some really big unresolved questions that we're trying to tackle in my lab. So one of the questions we're trying to figure out is what makes microbiomes diverse? There's a really great example that I'm showing you here on the slide that came from some early work of the human microbiome. So what I want you to do is look at, see those little pie charts that I have up there? And they're connected to different parts of the body, everywhere from your nose all the way down to your toes. And you can ignore the complicated microbiology names. Just look at all the different colors. Those different colors represent different types of microbes that are present on the average human body. And as you scroll across those different little pie charts, you can see that the microbes up in your nose, which tend to have a lot of those blue things, are very different than the microbes living between your toes. It's a really interesting pattern. We have no idea why. It's a really hard and difficult question to answer. Another important question to answer is, how do microbiomes assemble? So this is a really interesting question right now that we're all trying to figure out. So you can imagine when a car is being put together, there's an assembly line, right? And you have to put the wheels on and the axle at the right time. That steering wheel needs to go in at a particular time. Are there assembly rules, specific things that need to fit together in the right way inside of microbiome? And you can think about this personally. If you've ever had to take antibiotics, and your microbiome has suddenly changed, maybe you get sick and have symptoms afterwards, it could be because there are changes in the way that your microbial communities are interacting with each other. And again, these are really difficult questions that we don't have answers to. One of the big challenges of working in the human gut is there are thousands of bacterial species present, so it's a really difficult place to map out patterns. And finally, another really big question, and this is where there's a lot of money right now in microbiome research, is how can we manage microbiomes to improve the human condition? So there's a lot of companies I've listed right here that are specifically interested in developing probiotics for agriculture. Can we take microbes, spray them out into crops, and have higher yielding crops? And there's a lot of issues with trying to figure that out. So one of the big challenges, these big questions that remain and sort of are unresolved, is that it's really difficult to grow most microbes. So even if we went out to this New York City soil, which you think we would know really well by now, we've been trouncing over it for hundreds of years, if you tried to grow the microbes living in that soil in the lab, you'd only be able to grow about 2% of those microbes. We haven't figured out how to coax them to live in that complicated soil in the lab in a petri dish. So if you're only growing 2% of the microbes that are out there, it's really hard to figure out how they're all fitting together. Another thing is what we call irreducible complexity. So I like to put together puzzles that are like 20, 25 pieces with friends, kids. Trying to put together a 2,000-piece puzzle is a much more difficult beast. And so trying to understand how microbial communities that have thousands of parts work is much more difficult than simpler communities. And finally, one of the big challenges that's limited our ability to study microbiomes is accessibility. I have friends that study deep sea microbiomes. They have to get in a submarine and go a mile below the ocean to figure out what's going on down there. But actually, to study the gut microbiome is just as complicated. Trying to get inside of your intestine is not an easy thing. So these are some of the big things that have limited our ability to understand these big questions in microbiomes. So fortunately, there's been a really big push in the community, largely driven by President Obama's National Microbiome Initiative, where announced this past May, a big push has been to develop new model systems, things that we can culture, things that are relatively simple, and things that we can easily access to answer these long-standing and still open questions in microbiomes. And of course, you know where I'm going with this. Our model systems are these delicious fermented foods. So how many people love cheese? How many cheese lovers? Of course, everyone loves cheese, right? Yes, great. And my job is to get you hungry, by the way, for dinner. Surface ripened cheeses, these are things like brie and camembert, these fuzzy things that you see at Murray's or Saxelby's cheese in Essex Market. These things, if you look under the microscope, have a multi-species community of microbes living on them, just like the microbiome that you have in your body. My lab also studies kombucha. How many people have had fermented tea or kombucha? Okay, quite a few people. This is a rapidly growing beverage in the United States. $400 million a year of kombucha sold in the United States. It's fermented tea, so you actually make tea, add sugar, and then you add a microbial community to the top of that. And again, if you look under the microscope, you have a simple community of yeast and bacteria. And finally, we also study fermented vegetables in the lab, things like delicious kimchi and sauerkraut, which once again, when you look under the microscope, are teeming with microbial life. So at the end of the day, we're studying these systems because they're culturable, they're accessible, and they're relatively simple. We can easily go out to a cheese, to a kimchi, and to the other types of foods and grow all the microbes that are present, not just 2% of them. They're tractable in terms of their complexity. They usually have about 2 to 20 species, which again is a much simpler puzzle than a 2,000-piece puzzle. And the other really nice thing is that these things are widespread. They're essentially petri dishes that people are putting out around the world for us, little islands in South America, in Asia, in Australia, and all around North America and Europe that we can go out and study and then understand back in the lab. So at the end of the day, what we're trying to learn in these fermented foods is interesting for the food, but the nice thing is they're actually simple and relatable to other less tractable systems. So for example, what's happening in a jar of kimchi isn't that different in terms of the fermentation that's happening in your own gut. So if we can learn simple principles in these simple systems, we can then translate it out to the more complicated world. So how do we do this? What is our approach in the lab? Well, we do 3 different things. First, we go out and figure out patterns of microbiome diversity. We ask simply, how variable are microbiomes across these foods? We then bring the foods into the lab and try to understand the ecological and evolutionary processes that generates that diversity. How do microbial interactions, for example, microbes fighting with each other, drive these communities? And finally, we think about the molecular mechanisms, the ways that the microbes are talking to each other that might drive the patterns and the processes that we're seeing in these systems. So I'll just quickly show you what that looks like in the lab. Essentially, the first step is like microbial CSI. Instead of trying to detect a criminal out at a crime scene, we're trying to figure out what microbe is growing on a cheese. So we extract DNA from a sample, we amplify up particular genes in that sample that can then be put on a sequencer, and we can then match it out into a set of databases that can tell us what microbes are present. So again, it's just like what you might do with a criminal, except again, we're working with microbes. And at the end of the day, we get back these maps of the microbial diversity. And this is from our work with cheese. What you're looking at here is every column is a different cheese that we sampled. We sampled 137 different cheeses. And the different colors represent the different types of microbes on those cheeses. So you can see some of the cheeses have a lot of red, for example, and some of the cheeses have a lot of blue and green. Again, different microbes and different cheeses. The next thing we do is we bring the systems into the lab. So we make in vitro cheese, we make in vitro sauerkraut, we make in vitro kombucha in the lab, where we can make thousands and thousands of microbial communities to answer some of these big questions about microbiome assembly. And this is what it would look like. This is one of my favorite experiments we've done so far, and relatively simple. What we've done is we've combined different fungi with different bacteria and asked, how do they affect each other's growth? And what I want you to see is there's this one little circle that I have highlighted on the slide there, where a particular fungus is being killed off by a bacterium. And this is a novel way that bacteria are killing fungi. It may lead us to some kind of new compound that we could use to treat fungal infections. And that's all happening on your favorite wheel of cheese. We also get to work with chemists to figure out how these microbes are talking to each other. I like to tell people we're the microbial NSA, so we tap into the lines of communication that these microbes are using. And what I'm showing you here, again, like at a crime scene when they get out the ultraviolet lights and they try to figure out where the chemicals are, what we're doing here is we're getting out the chemical tools that we have to understand where different molecules are being produced by microbes. And microbes are really smart. We often think about them as these microscopic little bags floating around. You can actually see some of those colors I'm showing you have curves to them. And those are bacteria making compounds in a very specific direction away from a fungus. They're actually communicating in an opposite direction to avoid interacting with the fungus. So at the end of the day, we're trying to understand basic biology. We're trying to learn how microbiomes work. But we also care about food. And we get a lot of what I call Franken-cheeses sent to the lab. You never see these at the shop. They can never be sold. But cheesemakers always struggle with trying to understand the microbiome ecology as they're making their products. And so we can actually actively work with these producers to figure out some of these problems. And very recently, we've started at Tufts the Sensory and Science Center, where we can formally work with various industry partners. And we also work across other systems. So we're developing this in fermented foods, but we have a lot of collaborators. One of the great things about being at Tufts is there's a huge number of different people that we can work with, and it's really easy to collaborate. So everything from folks in the biology department working on honeybees and worms to folks over at the nutrition school trying to understand the role of microbial vitamin K in healthy diets. And finally, one of the challenges that we have as microbiologists is the ick factor, right? So many people, when they hear about microbes, they immediately get grossed out. And so how do we get the public excited? Well, we do it through food. So we have a website called microbialfoods.org where you can learn about the science of fermented foods. We've been in various podcasts, and we've been featured on numerous Netflix documentaries. We've also done things regionally. So we've had pop-ups for various food producers. We've done science museum exhibits, and we're going to have a farmer's market booth in the Boston area very soon. And the thing I've loved the most about this is using our systems to get undergrads excited about microbial diversity. Most microbiology classes focus on a single microbe throughout the entire semester. And what we've been able to do is use our systems in microbiology classes to get undergrads thinking about the complexity of microbes in the world around us. And fortunately, we have a fantastic undergrad here today who can tell you a little bit more about her experience with cheese microbes in undergraduate curriculum at Tufts. This is Kit MacDonald, and I'll let Kit tell you a little bit more about what she has to say.
I think Provost Harris is a bit taller than me. During my time at Tufts, I evolved a personal philosophy. A classic liberal arts student, I considered ways to find meaning among seemingly unrelated classes. Intro to Religions of China, environmental anthropology, photography. Truth be told, the philosophy is that nothing exists in isolation. This was a recurring theme in microbiology with Professor Wolfe. He ingrained in us that studying microbes in isolation would only give us a snapshot of the full picture. Every second of every day, trillions of microbes are interacting and cooperating and sparring on New York subway tiles, in transient puddles in the rainforest, and in between our toes. Thus, these microorganisms must be studied in multi-species ecosystems. Just as the fruit fly is a model organism for studying genetics, Professor Wolfe has named fermented foods as his model organisms of choice. And in doing so, he has entirely redefined the discipline of microbiology. My name is Kit MacDonald, and I graduated from Tufts this past May with a degree in biology. At this time 2 years ago, I had my future in academia all planned out. I'd start with a PhD in the evolutionary history of ants, and then somehow integrate my other passions for innovation and design later on. Then I studied abroad in Hong Kong. I took a course that introduced me to the exciting field of biotechnology business. When I returned to the Hill, I was uncertain of how my biology degree would now translate into a clear direction. That is, until I enrolled in Bio 106, Professor Wolfe's microbiology course. Now you must understand, this was no ordinary Microbio 101. In usual microbiology courses, students follow basic protocols to experiment with E. coli. Professor Wolfe, on the other hand, used a tried and true teaching technique. He motivated us with food, most specifically with cheese. We each got to adopt one for the semester. Mine was called Oma. As an undergraduate in a lab course, it's easy to go through the motions. In the hallowed, dank basement of Barnum Hall, you know full well that your experiments have been performed by thousands of jumbos past. However, the work we did felt really important because it directly related to the real world. We were the researchers at the forefront of the field. We were the first ones to look at the microbes in these cheeses. As a bonus, we got to eat our subjects of study too. Professor Wolfe frequently referred to us as future microbiologists and industry leaders. He suggested potential PhD theses and encouraged us to tackle the bigger questions. His vote of confidence in our abilities transformed the way we students approached the entire course and beyond. Now let me tell you, a block of cheese is much easier and appetizing to study than the space between your toes. And believe it or not, Some of the same bacteria take up residence in both places. Beyond working with cheese, we made sauerkraut in a highly scientific manner. We ate chocolate, a product of fermentation, as we watched Professor Wolfe featured in a Netflix documentary. Under the microscope, we hunted for ink-stained fungal arbuscules in leaks from his garden. We considered ways to apply light-activated proton pumps to novel technologies and how slime molds could be models for planning public transportation networks. And of course, we discussed which microbes would make great band names. Metallium, Toxothrix. But band names aside, Professor Wolfe totally sold me on microbes. They've been perfecting what they do for 3.8 billion years. The way to the future is working with them, rather than coating our world in hand sanitizer. Professor Wolfe introduced me to a biotech startup called Ginkgo Bioworks. They adopt the design principles of nature to create products using microbes. Since June, I've been working with Ginkgo's creative director and business development team. We introduced microbially produced molecules to new products and markets. Everything from luxury perfume oils, textile dyes, baker's yeast, to specialized probiotics and other custom strains for the Department of Defense. Biology, disruptive technologies, design, it doesn't get much better than this. Because nothing exists in isolation, including microbiology itself, this past fall, Professor Wolfe taught a course about food systems with both an anthropologist and a nutritionist. He Skyped in industry members for context, and I got to tell them how Ginkgo Bioworks technology is transforming the food industry. And after that Skype call, DeWitt McPhee's cheese fries certainly will never look the same way ever again. I've been able to combine my love for the natural world and the fast-paced field of biotechnology. This is completely owed to Professor Wolfe and his ability to share a passion for this brilliant, unseen world. His class is quintessentially Tufts. It characterizes so many of our experiences on the Hill. Reimagining ways to approach a liberal arts education, promoting students to take initiatives in unpioneered spaces, encouraging new ideas, however bizarre and out there they may seem, and creating unique opportunities for students. The ecosystems of Tufts are vibrant and thriving, whether they be networks of microbes on the Pres lawn or DeWitt McPhee, or a flourishing community of jumbos. Thank you.